Now we have Shawn up, our last panelist here, to talk about a very different use case with HIRO.
Great. So focusing on where to go with this idea that one is able to pool data together from different parts of different hospitals and put it together into someplace where the compute can happen, but that's highly secured. So I think there's also been this phrase that's been coined called trusted research environments, or TREs, and you'll hear about that. And it's very similar. I think the distinction is that in general, we're talking about something that isn't a permanent location for data. It's, and the reason that that's a problem is because when you, as a hospital, agree to put data into a permanent location that's outside your hospital, you're agreeing to trust, right, whatever that entity might be. But things can change in the world, as I think we've all seen over the past 4 or 5 years. And you often don't know long term what's going to happen to wherever that place is that you put the data. And so the idea is that with the ephemeral enclaves, like they have developed in Enact, that you can pull together the data from several different I2B2 sites on demand, if you want to call it that, and then assemble it for some time-limited period while the study is conducted. And then you can archive that, and it doesn't represent a risk to the patients or the world's changing circumstances. Now, in HIRO, we've been developing this concept of how to use data from I2B2 and OMOP and all of these different really significant collections of data to enhance the way we run clinical trials. And so we have to understand that in a leader like Mass General Brigham, you often are doing about over 2.6 billion, I think it's actually 2.7 billion now, dollars worth of research. And it does approximately 4,400 clinical trials. And so you might be like, well, that seems like enough. But the bottom line is that a lot of those clinical trials end up failing. And the reason that that happens is because we take on a trial when we know that we have the patients for that trial available at Mass General Brigham or not. And the only way to really understand that, and I think, Joe, there was a really great study that was done about 15 years ago. Aaron, where's Aaron? What was the name, what was his name? Oncology, sorry guys. Who? Did somebody say something? No, okay. No, it's a guy's name. Anyway, we're all getting old. The bottom line is that he showed that with I2B2, you could do a feasibility test and compare it to the success of the clinical trial. And if I2B2 showed that there were enough patients for the clinical trial available, it had about a 60% chance of succeeding, right? Which is better than 50 or 20%, let me just tell you, but not 100%. But if you did not have enough patients in I2B2, it had a 0% chance of succeeding. And that was the key, right? So before you do any clinical trial, you should really be doing a query in I2B2 or whatever system you have, but I2B2 makes it convenient in order to understand the potential success of the trial, because these numbers are very serious in that. And the 60% probably has to do with the fact that, as Griffin showed in a few presentations here, the claims data, which we often use as part of our queries, is highly inaccurate. And in fact, so using computed phenotypes in these feasibility trials would be a big win, right? It turns out that pharma loses many, many, many billions of dollars that would otherwise be going into building new drugs for folks by having these clinical trials fail 80% of the time, essentially. And I say that because, you know, a delayed trial to pharma is extremely significant. Their patents only exist for so long. And so if their clinical trial doesn't get completed, they lose, even if it eventually does get done, right? They lose all that patent time for the drug. Now, again, you know, we might be like, well, we're not very sympathetic. They're making millions of it, but they're not, right? Pharmas don't operate on enormous margins. And as I'm sure many of you are aware, they've been laying off a lot of folks because these margins have decreased, even though you think, well, they're making a lot of money for their drugs. Well, the problem is they have to spend billions of dollars on clinical trials at the same time. Okay. And then the 60% amendments is reflecting that the clinical trials have to be changed when they are not properly gauged to begin with. So HIRO has been developing this capability that solves some of these problems for pharma. It helps integrate it and kind of map out research strategies jointly with pharma. It streamlines study startup processes so that we don't waste time, and often 6 to 8 months is wasted. I think it was even in a presentation here, just getting the IRB through, right? Just doing the IRB takes that long. So if you can streamline that, you've just cut your trial by, you know, 6 to 7 months. And defining these automated cross-study workflows, and that's important. Then you want to take a look at and actually kind of do the retrospective studies and predict whether the clinical trial is going to work. And you can often do that in silico. I'm not going to focus on all the different mechanisms, but I am going to focus on one, by the way. Okay. But you can often do this in silico. And the way you can do this in silico is by putting the data that's relevant from the study, very, very similar to the way we discussed how ENACT is doing it and how Griffin's looking to have mass CPR do it, into an enclave with one extra step, and that is, because you now have that data in an enclave, what does it mean for the data to be an enclave? It can't go out, right? In other words, we're not giving the data out to pharma. We're only using it in this very secure, highly capable computational center. That means if we vet some people from pharma, right, we can let them into this, because the risk is that the data goes out and millions of people and hackers try to crack it, and they will, by the way, because even more so with AI, which can easily discern who a patient is if it's out in the ether. And unfortunately, many patients' records went out in the ether and go out in the ether every day because people are using AI and being like, oh, here's a patient, you know, what's going on? It all goes to Anthropic, OpenAI, Claude, and they say in their contract that they are going to use that data to train their model. So they are going to keep that data. And so if you send it to them, they are going to keep it. So that's why you keep the data in an enclave so it does not go out. If you're going to use an AI model, which we do a lot, we use local models, or a model that we have a BAA with that will not be able to have to worry that that data is going out to the internet and being used for all kinds of possible nefarious purposes. Okay, so that's the important thing about having the data in an enclave. You can bring some people from pharma in. It's okay, they're scientists. They sign all the confidentiality agreements, and they're just like us. And then they can bring their methods in as well and use their methods that they've developed for years in SAS and so forth, who knows, to do lots of great things in these enclaves using their methods. So the key here is the way that we can enable a better relationship and success with pharma, working with pharma, is to have these enclaves in place at our institutions. And then you can do prospective studies, understanding how you can develop adaptive recruitment studies, strategies, and fast-tracking contracting and IRB approvals for that, because you have evidence now from your enclave. So this data enclave, as we've kind of, just to kind of formulate it in your head, right? It's this lockbox environment. And researchers then develop things like AI. They can use all these different tools, and oftentimes containers bring in even proprietary, I will say, pharma algorithms to be run. But all this then can be run very, very securely in this environment. And the data can be created from common data models so that these can be run reproducibly, and we can run lots of the things that have been promised basically in this conference using this kind of approach. And there's lots of different kinds of data. Don't, kind of think about, the usual ones: demographics and diagnoses and procedures and lab results, but also prescriptions. Habits is very important, by the way. In the biobank, we actually collect habits from patients, smoking and drinking. Very important in our society to know about that, as well as drug use, illicit drug use. And the bottom line is that that's an important factor. Clinical notes and reports are really important these days with AI. They provided a whole new window, and we're going to talk about that in a little tiny bit. And then telemetry, specimens of biological specimens. That's supposed to be a syringe full of blood, by the way. Imaging, patient-reported outcomes, and genomics. And we've talked a lot in this conference about improving data through computed phenotypes. And the main thing we can do here with these different kinds of enclaves is create inside them a registry that this gets maintained for 1 or 2 years. Not forever, but for 1 or 2 years while pharma is doing a project. And it can do this proactive natural language processing and automated learning and clinical validation like we've been talking about. And I want to bring out another feature that the team has been working on with generative AI. And it's going to combine several things that we've discussed. It's a pipeline to determine something as important as what are all the stages of cancer that a patient has gone through over the past 15 years. And that means they'll go from stage 1 to stage 2 to stage 3, maybe to remission, then maybe back to stage 2, and then unfortunately, to metastatic, stage 4, and so forth. And the reason that's so important is because, one, it's the first step in looking at the outcomes of the patient. And two, pharma often wants to know how many patients do you have in stage 3, right? Or how many patients are developing going into stage 3 a month overall, so that they can calibrate, oh, then we're going to try this oncology drug, right? This new kind of chemotherapy. So believe it or not, that's extremely difficult to understand, to discern. Now, Epic actually does have a smart form where you can fill out all the staging information for the patient. And it's very nice, actually. It goes into a nice structured database. About 10% of the doctors use it. 10%, what we found. I said 10% use the smart form in Epic. Now, That's simply because I will tell you, it's really hard to fill out. And so, and the law requires you, I'm not sure it's the law or it's the cancer, thank you, yeah. That you have to say somewhere in the note with a date what stage cancer they are entering, right? And if they enter another stage, you have to put in the note, they're entering stage 2 on this date. But that's the only thing that's required, and you will find that. Okay. So 100%, presumably, of staging can be done that way. But you've got to find it in all these notes. Now, we've been talking about large language models. Can it find it? Sometimes, actually. I mean, pretty often. Not, well, anyway, pretty often. But here's the problem. We have we have 250,000 patients with cancer. I'm sorry, or happy to say that they're coming into Mass General Brigham to get treatment. And they have hundreds of different kinds of cancer. So, or 72 kinds of cancer as Mass General Brigham classifies them, by the way. Are we going to have a large language model look through? Each one of these patients has about 1,000, at least 1,000 notes, at least. So we're going to have them look through 250 million notes. Did I do that calculation right? Yeah, okay, 250 million notes, right? And the answer is, well, that would take about 30 years. So no. Now you could say, well, do it in parallel. Great. And that's going to take about $10 million like a month. Okay, so that's not going to really work. But change your thinking. Think of the large language model as the human. What would a human, how can it create something which we've been talking about here, but could become a closed loop computerized process? All right. So in this diagram, we're looking at how is it that we're going to extract cancer staging information in a timeline. I call that a timeline when that goes from one stage to another, then remission. Timeline for each cancer patient. And we want to automate this process. So what we have to do is figure out who has some kind of cancer. And all the different cancers, by the way, have different stage nomenclatures. So you actually have to do this for each cancer. But you want to figure out how many patients actually have the cancer. Well, we got computed phenotyping. Great. Now remember, when you do computed phenotyping, we do need to have a human check whether they actually have the phenotype for some small set of patients, 200, let's say. But 200 patients to actually check the charts, to do a chart review, will take a month, maybe 2. And I just said we have 72 cancers, so we're talking 5-6 years, right? Have the large language model do the chart review on 200 patients. That's not too bad, right? It'll take a day, right? So we're talking a day for each phenotype. So we got 72 days of large language model time. That's pretty tractable. So that's the first part, find out who has the cancers. Second part, we got to read the chart and figure out who has the stage annotation, right? And it's not only in the ambulatory notes, it's in the pathology notes, right? Because there's both clinical and pathological staging. So again, we're back to our millions and millions of notes. But how about this? A human would say, oh, I'll use a regular expression. And we're like, regular expressions, those are horrible. And they are, I'm going to say. They're really tough to work with. But, they'll do the job if you let the large language model figure that out. And let the large language model figure out if it did it right or not by giving it a few examples that it came up with, right? So let's say it gives you give it, say, find the stage in these notes, and then it looks through it and it says, I found it in this, using my regular expression, I found it in this 500 notes and not this 500 notes, and then let it read actually the notes, the large language model actually read the note. and say, oh, I missed it in, 8 cases, and then the answer is fix your regular expression and do it again, and check yourself again, and then keep doing that. And that is a closed loop system, no human. That's what we have in the middle here, and that is what is really enabling us to use large language models to do 200 million notes. Regular expressions run 200 million notes is no problem. A couple of hours, right? Then you got to put all that data together because you have to fix things like, well, you found that stage 1 started in January and it ended in February, and then stage 2 started in January. Well, wait a minute, it should start in February if the stage 1 is, okay, so something's got to fix that. Again, that's normally a human. Let a large language model and the third thing do that. So now we have a large language model figuring out who's got cancer, what kind of cancer, doing the staging, and fixing up all the staging to make it kind of logically coherent, all in a closed loop system. And that's the work. So we have Vivian Gaynor, we've got Victor Castro, I don't know, is Nitch still here? Nitch is here, Nitch Wadanasan, Mike Mendez, Baswati, Hikyong, thank you. Anyone I missed, Vivian? I said Victor. All right, that's what the team's doing. And here's the result. You get all these cancers, and we haven't done it all yet, so but I have the one we have. And this beautiful I2B2 laid out structure that can now be used in the enclaves with Pharma. All right, so that's the kind of thing that enables a registry to be created in the enclaves in HIRO. You can extend this concept to outcomes and many other things, creating these multi-modal data applications. With that, we can actually create this digital twin concept, right? You can really put patients completely together in a way that's never been possible without this kind of generative AI insertion, to really kind of focus on creating digital twins, which can actually allow you to do even more amazing things, right? Like, if you have a cancer patient, what's the worst thing, if you're going to be a cancer patient in a clinical trial, what's the worst thing that's going to be able to happen? You're going to be in the control group, right? Who wants, I mean, you're there because you want a life-saving treatment because you've probably failed most other treatments, and now you're going to get put in the control group. Let the controlled group be digital twins. Let them be digital twins. So that's where we can go with data-only trial arms. We can do a lot of research acceleration using these kinds of approaches to accelerate and do much of what we need to do in silico. And finally, advance the way we do translational medicine in the hospitals in a way that we can go hand in hand with pharma to work on things that ultimately are one of the greatest benefits we can provide to our patients. New drugs, right, to treat their conditions. Okay, so this is kind of the little HIRO team. We have been looking at putting this same program into other hospitals. I've talked to many of you, but anyway, here's how to contact us. Thanks very much.